is that for fluorescence, you can just track your cells for a couple of days, I would say up to 3 days, but not more than that. In bioluminescence, you can track them for months, for years, as long as they are surviving. So this is of course, it's not using the same equipment. It's still optical imaging, so it's a similar instrument in the way that you need a CCD camera. But since bioluminescent signals are very weak signals, you need a very efficient CCD camera. So we are cooling down our CCD camera down to -90 degrees to avoid the background noise. We call it electrical noise from the camera. So we are cooling our CCD camera at a very low level in order to be able to pick up those small amount of photons. And the other really nice thing with bioluminescence is that there is no background. There is no autofluorescence from the tissue, from the skin, or anything. So there is no background, even if it's a small amount of light in the middle of nothing, it's pretty easy to detect. I'm used to say that bioluminescence, you are looking at stars in the sky during night, so you can perfectly see all the stars if there is no cloud. And fluorescence, you are looking at the same stars, but during the journey, when there is the sun which is shining. So of course you are using 800 and 750 nanometers, and in this specific wavelengths, autofluorescence is not that high compared to 680 or even other range like in the green range or blue range. But still, you already have some bad photons coming from autofluorescence. So you will see that in terms of in bioluminescence, your images will be way more clean. It's going to be a cleaner imaging compared to fluorescence. Yeah, so it's a different strategy. So what you will need to do is to transfect your cells in order to integrate a piece of DNA coding for luciferase into your cells of interest, such as your stem cells, for example. And the substrate of this enzyme is called deluciferin. And it was expensive long time ago. Nowadays, since it's used by many people, the price brought down quite a lot. So you can buy a vial with a powder. You just need to resuspend the powder and to do an IP injection. This substrate will diffuse into the mouse, and you will see light emission where the luciferase is produced, basically in your stem cells or in your cells of interest. So this is how it works. It's really a biological reaction, enzyme plus substrate. And this enzyme is coming from shrimp or firefly or worms, so it's not present by default in murine cells or in human cells. Okay, so maybe we can start, because it's already 16, so I don't know. Maybe we can start and people will join during the call, or let me know, Kalma, what you think is the best to do. Should we start or not? I think you are mute, Kalma.
I don't know what could happen with the rest of the people, even though from Teresa de Francisco, who was she was agree with doing this, and I really don't understand what could happen. But in any case, something. Yeah. Yeah. I don't know if Christian, can you hear us? Because we have another person connected.
I can see the name, but
Anumat.
by default is also muted. I can see that the microphone is off, so not sure if he has a microphone with him or not.
Okay, in any case, I think if Carlos have any interest, specific interest, and he wants to talk about what could be the advantage for him working with an instrument different. Yes, but I haven't neither.
That's fine, as long as you have headset and you can hear.
If they want to know more a little bit about luminescence, we can make some short presentation for them because they have time for us, and we need to know.
Yeah. Also, this support will be available. We can also record if you want. So there is many different things we can do. We can just send out my presentation after the call so that they can review it if they want. We can also register this seminar if you want. So just let me know what you want to do and we will do it. Do you prefer to register or to send the presentation afterwards?
Yeah. Carlos, do you think that luminescence could be helpful for your daily work?
Yeah.
Newton. From Bill. Yeah.
Yeah. The new system you had, the university can do bioluminescence, can do both fluorescence and bioluminescence. It doesn't have the highest sensitivity you can get because there is better instruments, but it can do bioluminescence. So it could be also a good start, I would say, if you want to just see how your cells behave with bioluminescence. So as I said before, the really big added value, especially when you are interested in stem cells, is that bioluminescence will give you viability. So what is the percentage of living cells? If you inject, I don't know, 1 million cells, how many of them are going to die? How many of them are going to stay alive? And you will be able to do domiciliation studies. Where are they going inside the animal? And this as long as they are living. So you can track stem cells for months, for weeks, for years if they are staying and living there. The other added value is that since it's a gene reporter, if your stem cells are dividing, you will also see an increase of the signal. So the quantity of signal is completely linked to the intensity of the light. Yeah. Yeah, the idea is to make them as stable as possible. So there will also some development in that people has to do on this side, but it's not a transient one. We want to do permanent transfections. In order to do that, we are using very strong promoters, such as ubiquitin promoters, or well, we have a couple of them, CMD promoters, for example. And well, of course, you have to make sure that the signal stays stable over the time. But I will show you some experiment active. I know that I have one in my presentation using human stem cells. It's neural stem cells in a model of spinal cord injury. And people are using those human stem cells in order to recover from spinal cord injury. So I will show you that they are checking viability, and they are also just using the stem cells to make the mouse move again, because they were, since the spinal cord was broken, they could not move correctly anymore, and they can benefit of these human stem cells in order to recover from their pathology. Okay, so maybe you can start. I think otherwise we are never going to start. So let's start and let's see if some people will join. They will join during the course. Whatever, I will send my my presentation to people who want it. Okay, so let's share my screen. Okay, you should be able to see my screen right now. Just going to put that one here. Okay, let me know if it's fine for you.
Yeah.
Okay, perfect. So let's go on. I will just start with a few basics things about bioluminescence first. So first of all, just to talk about all the different imaging modalities. They can be split in two different groups, functional modalities and anatomical modalities. In the functional modalities, you will find nuclear approaches and optical approaches, like SPECT and PET for nuclear, and fluorescence and bioluminescence for optical. And for the anatomical modalities, you will find, of course, MRI, micro-CT, and ultrasound. So in order to be able to make some images, we need some reporters, and in bioluminescence, we are using a gene reporter, so it's a gene reporter technique. So you need to transfect your cells and to include this piece of DNA coding for luciferase into your cells of interest. Then those cells can be injected into a mouse, and when you inject the substrate, you will see a reaction between the luciferase, which is an enzyme, and the substrate, which is called deluciferin. So this enzyme can come from many different species, and I will show you that in a minute. So for the fluorescent approach, you already know it. You can use synthetic dyes that can be graphed to molecule of interest, such as antibody, or you can find lipophilic dyes that will stay inside the membrane of the cells. But as I said, you will only be able to follow cells for a couple of days, but not for weeks or months. On the other side, you can also use a gene reporter technique in fluorescence, but you will achieve a way lower sensitivity compared to bioluminescence. We used to say that between fluorescence and bioluminescence, the difference is around 1,000 to 100 times more sensitive for bioluminescence compared to fluorescence. So we are still facing a big challenge when we are using photons inside a living animal, because as you can imagine, photons are easily absorbed and scattered in soft tissue. And also, when you have 2D imaging systems, the signal you will collect will always be the signal on top of the mouse, which means the signal that can escape from the mouse. So also this signal and its intensity specifically is dependent on the depth. So deeper the signal is, lower will be the amount of light you will be able to measure. Another phenomenon is autofluorescence. So when you are in a fluorescence mode, you have lots of bad photons coming from the skin, coming from the food. When you are in luminescence, autoluminescence is almost zero. So just to emphasize on diffusion, this is just, it's a glass of water. So if you have just pure water, the medium is not diffusive. So you can see that the laser pointer can go through it without diffusion. But if you put few drops of milk into this glass of water, you can see what diffusion does. Diffusion will just make the laser pointer not as sharp as it could be, and you will see the light starting to diffuse inside this medium. Now, to talk about not diffusion but absorption, light is easily absorbed in soft tissue, especially by blood, by hemoglobin. So you can see here different spectrum coming from different components from soft tissue. You can find the proteins, you can find water in the black line, collagen, blue line, but all those components are not really the ones that are making problems. The main one which is making problem is HbO2 and Hb, so hemoglobin and oxyhemoglobin. So it's two different spectrum here. If you fuse both spectrum together, you will find a very high spectrum, which is the main problem with light absorption. So we also know that higher than 650, hemoglobin is absorbing less compared to 300 or 400 nanometers. So as much as we can, we try to stay in this near-infrared optical window, 650 up to, let's say, 800 nanometers, because there is not much fluorescence or bioluminescence reaction higher than 800 nanometers. So in fluorescence as well as in bioluminescence, we will try to stick in this optical window as much as possible, just to avoid light absorption by hemoglobin. So this is just a quick comparison between bioluminescence and fluorescence. In bioluminescence, it's just about few photons, but since we don't have any background at all the signals are pretty clear to be detected. So this is just tumor cells injected into the pore. In fluorescence, this is one of the worst fluorescent images you can get, but anyways, just to give you an example, you can see some background from the instrument just by light reflection or leakage around the filter. You can see also some fluorescence coming from the full animal. This is the autofluorescence from the skin. And you can see some signal here in the scapular area of the mouse, but the real signal, the real fluorescence is just here. So it's not that easy to find the real fluorescence. So what we need in bioluminescence is a sensitive camera, as sensitive as possible. And in fluorescence, it's not really a matter of camera sensitivity. It's much a matter of having the right tool to be able to remove the bad photons from the good photons. So just about the bioluminescent reaction. So as I said before, it's a reaction between an enzyme, a luciferase, and the substrate. So what you will first need to do is to integrate this vector into your cells of interest. So people are using lentivirus to transfect their cells or adenovirus, and they're using this kind of vector. So in this vector, you can find the piece of DNA coding for the luciferase and an upstream element, and some selection marker just to select the cells that are perfectly integrated this vector and the one which has not integrated the vector. For the bioluminescent reaction, you need to inject the substrate, the luciferin. This injection is done sub-Q or IP, and you will see that in presence of luciferase as well as ATP and magnesium, you will find the first reaction. D-luciferin will be transformed into D-luciferyl adenylate, and in presence of oxygen, D-luciferyl adenylate will become oxyluciferin, which will emit light spontaneously. So you can find many different luciferases. They are not all equivalent, but you can find luciferase coming from firefly, from chimpanzee, coming from shrimp, coming from beetles, coming from worms. And as you can see, they have different wavelengths. So of course, we prefer the ones that are expressing their light in the red range compared to the ones that are expressing light in the blue or green range. Also, when you have a deeper look into those luciferases, they have different cassette size. So some of them are better for example to put inside a virus. Some of them are better to put inside eukaryotic cells. And also their light emission can be different. Some of them are just emitting a flashlight, so it's like a peak, and then it starts to decrease. And some of them are more glow, so they are emitting a kind of stable light for a couple of time. Here I have just used different spectrum from different luciferases. So you can find vanilla luciferase here in blue, the luxoperon for bacteria, also in blue, twig bitter green in the green range, and one of the most used luciferase called firefly luciferase because it's emitting light in the red range. Twig bitter red is also emitting light in the red range. So we will try to use this one as much as possible to avoid what you can see here in gray, which is light absorption by hemoglobin. We can see that there is a huge absorption in the green and blue range compared to the red range. Okay, just to talk about sensitivity, this is the kind of sensitivity we can achieve. Those cells were T cells. T cells were isolated, transfected, and reinjected sub-Q into a mouse. So you can see that we can just detect few cells, like 3 cells, 30 cells, 300 cells in a mouse. So here it's not really deep in the mouse, it's just sub-Q, so it's easier to detect. But we used to say that we can detect down to 200 cells wherever the cells are located, in the lung, in the liver, or whatever the localization is. 200 cells could be detected if they are bright enough, of course. Because it's not only a matter of sensitivity of the camera, it's also a matter of the brightness of your cells. We also have publication with just one single cell, 41 breast cancer cells injected sub-Q. Okay, just to give you a quick difference between 2D bioluminescence and 3D bioluminescence. So in 2D, we are just measuring the photons that can escape from the mouse. So we are measuring a circle of light on top of the mouse. But when you have this kind of circle of light, it can come from 2 different events. It can be a very intense source, which is very deep, and by diffusion, we give you this pattern of light on top of the mouse. Or it can be something which is way less intense, but with a bigger size, just sub-Q. In both cases, you will get the same task of light on top of the mouse, even if it's 2 different events. If we want to go further and to have an idea of the concentration, the size and the depth of the signal, we need to do tomography. So we need to do 3D in order to understand and to correct for absorption and diffusion of light. So this is just a quick example using just a phantom with a bioluminescent stick inside it. We are just collecting images at different wavelengths. We reconstruct the envelope of the mouse, and then we have an iterative algorithm that will relocalize the signal in 3D. So now we can not only measure the signal that can escape from the mouse, we can measure the real intensity coming from the signal where it is located. So this is an in vivo acquisition of a mouse. So I don't know why when I'm using Teams, I cannot. Oh, okay, there I go. So it's CAR T cells. So CAR T cells that have been transfected and injected IV into a mouse. We have a 3D system that can also create anatomical maps using microcity. So it's a microcity embedded into our optical system. And you can see in this case that the localization of the CAR T cell after being injected IV are localized in some of the lymph node, but most of them are localized in the spleen. And I have added the spleen from a mouse atlas here, just to clearly show that the presence of the CAR T cells are in the spleen here. So we have a clear understanding of the localization of the cells in 3D in the mouse. Okay, just talking about application now. So you can monitor many different events in a mouse. So you can monitor disease progression. I will show you images and slides of tumor development and growth, of drug efficacy studies. You can also study pathogens like bacteria, viruses, and also parasites, fungus. We can also have a more cellular component view. So you can do homing studies, migration studies. You can study stem cells, so engraftment, hematopoietic stem cell which just divide over the time and you will see an increasing signal. You can check for immune response. And we can also have a look at a more cellular and molecular components by selecting not a promoter which is activated all the time, but using a promoter which is specific to certain pathways. Start with the strategies of bioluminescence using a constitutive promoter. So do you remember the vector I was showing you containing the cassette for the luciferase gene? We have to select an upstream element. So we have to select a promoter for this luciferase gene. When people are expecting to follow cells, they are basically using a very active promoter, so like ubiquitin or CMV, which is always active, always on. So luciferase is always produced into cells. So we can detect cells, and not only detect cells, we can also check for their viability, because a dead cell will not emit light anymore. So what you have to keep in mind is that the collected light is proportional to the number of living cells. So then you can follow cancer cells, leukocytes, stem cells, pathogens, and I will show you examples, for example, for cancer cells. So this is basically cancer cells that have been injected into the lung. So it's basically 2 mouse with lung tumors, and we can clearly detect this tumor in the lung here. Also detect other type of cells, such as leukocytes, for example. So this is T cells that are present in the lymphoid organs, such as lymph node, the liver, and the spleen. So we can detect those T cells here in mouse. We can also detect stem cells. So this is hematopoietic stem cells that have been injected into a mouse that has been fully irradiated to kill all the bone marrow. And you can see that mouse that have been grafted with stem cells will survive. And you can see the localization of the stem cells, and you can also see where they are migrating, and also the fact that they are dividing over the time. If I didn't show the control group here, but when you look at the control group, they are all dying, I think, between day 6 and day 9, they are all dying over the time. And you can also track pathogens. So this is a bacteria that is emitting bioluminescence. And we used barium sulfate to highlight the cecum and the colon. And you can see the perfect localization of those pathogens. So it was bacteria inside the cecum of this mouse. I was talking about stem cells, and I was talking about this human neuroepithelial stem cells. And this is exactly the slide I was talking about. So on the left-hand side, you can see that people are just checking the survivability, so the viability of their cells over the time. And you can see that after grafting 100% of their cells week 1, they are checking every 2 weeks the quantity of light which is emitting from these mice. And you can see that after 2 weeks, they lost 60% of their cells, so 60% of them just died. But they were quite happy to see that even 5 weeks after injection, more than 20% of them were still alive. So after being sure that their human stem cells could survive into the mouse, they also checked the efficacy of these stem cells after transplantation. So here you can see a slide showing let's say, a value which is based on the mobility of the mouse. So if the mouse can move, then you will see that the score will be high. If the mouse cannot move, the score will be low. So here you can see the control mouse. So control mouse, they were suffering from spinal cord injury. So basically, the spine has been disrupted, and they could not move that much. So in the control group, you can see that the mouse are not moving very much. So the BMS score is about 2. And in the group, which has been injected with this self-renewing neuroepithelial stem cells, you can see that they will recover from this spinal cord injury and their mobility is increasing. And just to make sure that this increase of mobility is produced by the neuroepithelial stem cells, people were using the diphtheria tetanic diphtheria toxin, and this diphtheria toxin will only kill human cells. So it will have no impact on murine cells, but it will kill the human cells. And you can see that after using this diphtheria toxin, human cells are killed, and then the mouse is losing the recovery she got from the neuroepithelial human cells. So you can also see the localization, the quantity, the viability, and the impact of these stem cells on the function of the mouse. So if we can detect cells, leukocytes, stem cells, pathogen, or cancer cells, we can monitor disease progression. And this is what people are doing most of the time. They are just injecting tumor cells that are expressing bioluminescence, and they will monitor the tumor growth over the time. And basically, the advantage and the benefit is that what people were doing in the past is using a caliper, but the problem is that when you are using a caliper, you can only measure tumors that are sub-Q. If you are in an orthotopic brain model, you cannot use a caliper anymore. And also we can see that measuring bioluminescence, even if there is no mass, we can still detect and quantify the number of living cells. So we have an output way earlier compared to the caliper, which is only able to measure mass of tumor, but not spread tumors, for example. So here, just to say that we can monitor tumor cells over the time. And we can follow them until you can see some metastasis. Here, for example, it's a breast cancer model. So breast cancer cells have been injected into the mammary fat pad. So it's an orthotopic model. And you can see that this primary tumor will develop metastasis in the lung. And you can see that all the small signals from here are different metastases. So we just remove the lung from the mouse to confirm that we have a really good delineation of all the metastases inside the lung, even ex vivo. So we took this lung, we made some staining, so hematoxylin eosin staining, just to confirm the presence of metastasis in the lung. Okay, so if we can monitor disease progression, we can also use different drugs in order to see if we can kill the tumor cells. So this is just an example using two different treatments, 5-FU and mitomycin C, to kill tumor cells. And this is the control using just saline, so NaCl. We are measuring bioluminescence week 1, week 3, and week 5. So again, the signal is coming from tumor cells. We can see that in the control group, those tumor cells are just dividing, and the mouse will finish will finally be dead. And using a 5-FU treatment, one treatment between week 1 and 3, two treatments between week 3 and 5, you can see that you can not really treat the mouse, but this is a green curve. You can reduce the speed at which cells are dividing. They are still dividing, they are still growing, but slower than the control. And using just mitomycin C with just one injection between week 1 and week 3, and a second injection between week 3 and week 5, you can see that you can somehow cure the mouse. You can completely reverse the fact that the cells are not increasing over the time anymore. They are just being killed more than they are dividing. And you can see that the curve is going down. Okay, so let's just show you another drug efficacy studies, but this time I'm not following tumor cells, I am following bacteria. So it's a Streptococcus pneumoniae intratracheal drug efficacy studies. So basically, those Streptococcus are emitting bioluminescence. So you can see the presence of those bacteria in the lung. And we have injected 10 to 6 CFU. First line, we are not doing any treatment, and we are measuring the different, so the mice at different time points. So it's in hours, so 0 hours, 24 hours, 26, 44, and 48 hours after injection. And we use two different treatments here, 1 milligram per kilo of amoxicillin or 5 milligram kilo of amoxicillin. So same compound, but two different doses. And you can see that by just using 1 milligram per kilogram of amoxicillin, you can really reduce the infection, so the lung infection. And by using 5 times more amoxicillin, you almost kill all the bacteria. You could not detect any signal anymore at 26, 44, and 48 hours after infection. So cell homing, if you want to follow stem cells, for example, this is a study using two different cell lines. So one cell line is a tumor cell line, so it's 41 breast cancer cells, and those tumor cells are tagged with renila luciferase. So a luciferase which is emitting in the blue range. We know that it's not perfect for in vivo imaging, but since it's tumor cells, and we know we are going to get lots of cells, it should be fine. We should be able to detect those cells. So you can see 41 breast cancer cells, they have been injected in two different localizations. Again, it's an orthotopic model, it's in the mammary fat pad. So you can see the development of those tumors inside the mouse. And we used a second cell population, which is a mesenchymal stem cells, that were tagged using firefly luciferase, so emitting in the red, which is way better to detect smaller amount of cells, because we know that the quantity of mesenchymal stem cells is going to be way lower than the quantity of 41 breast cancer cells. So what we can see here, it's exactly the same mouse, and we are just following two different cell populations inside the same animals. So we can see the localization of the tumor with the first line, and in the second line, you can see that the mesenchymal stem cells will first stay in the lung half a day after injection, as you said, because the lung is the first filter. So this, when you inject cells in IV, it will go to the lung first. And then you can see that there is a specific tropism, and the mesenchymal stem cells will move from the lung without any specificity. They will move with a specific, let's say, with high specificity to the tumors. And we can clearly see a good colocalization of the blue bioluminescence and the red bioluminescence coming from two different cell populations in the same animals. And you can see that 12 days after injection, mesenchymal stem cells are not really dividing over the time. So in this mice, for example, you can see that we have a lower amount of stem cells compared to day 6, and it's the opposite for the other mice. We have a higher amount of stem cells at day 12 within the tumor, and also because the tumor is way bigger in this mouse compared to the other one. So when we are about to detect two cell population inside one animal, we are facing a huge problem when it's not at the surface of the animal. So you can see in this application, both signals are coming from really surfacic localization. So we are not really deep in the animal. But if we are going deeper in the animal, you will see that the green luciferase or the blue luciferase will be fully absorbed by hemoglobin. And the only part of this luciferase which is going to be collected will be completely overlapped with the red luciferase. So this is the two different luciferase spectrum. This is what you will get in vitro, so in a well plate, because there is no absorption by soft tissue. So in vitro is pretty easy to separate both cell population based on their spectrum, but in vivo it's almost impossible if the signal is deep in the mouse. So people were developing new luciferase called Firefly mutant. So one is called Firefly Luc green, the other one is called Firefly Luc red. So it's two different mutants of the initial Firefly luciferase. And they also played with different substrates. So they decided to choose the most optimal substrate called infraluciferin here. And if you have a close look at all the spectrums, you can see that if you're using the normal substrate, the luciferin, the green firefly is expressing in the green range, and the red firefly is expressing light in the red range. But what we want is to avoid emitting light in the green and blue range. So people find out that using infraluciferin, the light emission from the same enzyme were shifting in the red range. You can see that just by changing the substrate, you can see that the green firefly luciferase is now emitting at 680, and the red firefly luciferase is now emitting at 740, 720. So just to show you that if you are using those two mutants with infraluciferin, you can easily distinguish two cell populations even if they are deep in the mouse. And they have used those two different mutants in tumor cells, so it's basically RegB lymphoma cells. Here on the left-hand side, they have transfected those RegB lymphoma cells with just the green luciferase. Okay, and this is what they could detect with different filters. The idea of using different filters is to be able to plot the spectrum of this green luciferase. And they have transfected RegB lymphoma cells with the other enzyme, the firefly look red. And you can see also, using different filters, you can draw the spectrum of this F-look red. So since they were using infraluciferin, you can see that we have one spectrum around 680 and another spectrum around 720. Perfect, so this is subcutaneous. What happens if you inject the same cells, so the green one or the red one, IV, so in the systemic compartment, you can see that there is a distribution of the cells. They are, in the end, in the knees probably and in the liver, as well as in the salivary gland, I guess. So you can see those localization. Whatever, if they are green or red, you can see that this is way deeper localization, especially the liver, and we can still discriminate them based on the spectrum. So this is the green spectrum, still centered at 680, and the red spectrum centered at 720. Another model, intracranial, so the same cells were injected into the brain. Some of them were green, some of them were red, and again, you can completely discriminate between the green and the red. So now the idea is to inject the tumor cells not in different animal, but in the same animal. So here they have done a study where RegB lymphoma cells were expressing the green luciferase, and they have also injected human CAR T cells that are specifically expressing CD19 just to target those RegB lymphoma cells. Both cell populations were injected, and we are using spectral mixing, so we are using different filters in order to separate the two populations based on their spectrum. So we have 4 animals here that have been injected with the tumor cells. The one on the left has only been injected with tumor cells, but it has not been injected with CAR T cells. So just one cell population for the mouse on the left, and the 2 cell population for the mouse in the middle. If you look at the tumor-only mouse, you can see that tumor cells are growing over the time, normal, there is tumor growth. So this is the lymphoma which is growing over the time. And there is no CAR T cells signal because there is no CAR T cells injected into this animal. When you look at mouse that were injected with both the tumor cells and the CAR T cells, you can see that the tumor cells are not growing as they should because of the interaction with the CAR T cells that are killing the lymphoma cells. And here in red, you can see the signal coming from the CAR T cells, which is increasing over the time, meaning that the CAR T cells are going on the tumor side over the time. So it's not because they are dividing, it's because they are migrating from the liver or from lymphoid organs to the tumor. So if you look at the images, this is just the green image. So on the grid, on the green image, we can only see the lymphoma cells. On the red image, we can only see the CAR T cells. And on the composite image, we can see both signals. So we can see the green signal coming from lymphoma and the red signal coming from the CAR T cells. And when they are overlapped, it turns yellow. So you can see some yellow signals here, 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 and here as well. Okay, just another approach of using bioluminescence. So you can use bioluminescence also to perform kind of biodistribution studies of, for example, nanoparticles. So I wanted to show you this study where people developed lipid nanoparticles, okay, and they tuned the composition of their lipid nanoparticles with more or less cationic lipids. And you can see that when the lipid nanoparticles are heavily cationic, they are targeting the lung. When they are half-half, they are targeting the spleen, and when they are not cationic at all, it will target the liver. So they developed 3 different lipid nanoparticles, and in order to be able to visualize where those nanoparticles are going, they just inserted into these nanoparticles mRNA, so the luciferase mRNA. So when those nanoparticles will go into cells of interest, of course, the membrane will fuse with those local cells, and mRNA will be traduced into the protein, so the luciferase, and you can detect light coming from those nanoparticles. So cells that have integrated those nanoparticles. So you can see that in this mouse, if you inject the red one, you can see only luminescent signal from the liver. If you put the liver ex vivo and all the other organs, you will see that only the liver will emit light. Same for the spleen. Same for the liver here. So this is lung, excuse me, and this is the liver. They also played with concentration. So they changed the concentration of mRNA. If they increase the concentration of mRNA, they are able to deliver more mRNA luciferase into the cells of interest. So there is a dose response as well. And they also check over the time how long does it take for the nanoparticle to achieve their target. So you can see that for the lung, for example, just after 3 hours after injection, you have the highest possible signal. Same for the spleen. 3 hours after injection, you can see that you have already the highest quantity of nanoparticles present in the spleen. But for the liver, it's slightly different. For the liver, you have to wait 8 hours to have the highest amount of nanoparticle delivered into the liver. So it's a nice way to just deliver compound just based on the quantity of permanently cationic lipids. They can really target different organs. And you probably know that by default, when you are using any kind of nanoparticle, it will target the liver because the liver will just detoxify the organism by trapping those nanoparticles and making some degradation in hepatocytes or lymphocytes. So this is it for the constitutive promoter, so the approach where the luciferase gene is always expressed. It's basically to follow cell populations. But we can also think about specific expression of luciferase. And in this case, we are going to create transgenic cells or transgenic animals that will only emit light under certain circumstances. For example, there is a mouse called the NF-kappa B mouse. This NF-kappa B mouse is a transgenic mouse. Whenever there is an inflammation anywhere in the mouse caused by whatever, the mouse will emit light. Same for the GFAP mice. So the GFAP mouse is a transgenic animal, and GFAP stands for glial fibrillary acidic protein. Whenever there is damages on the neurons, the astrocytes will synthesize the GFAP protein, and when the GFAP protein is synthesized, luciferase is also produced. So we use this GFAP mouse in many different applications, especially in the brain, to see some damages done on the neuron. If you are more interested about neuroangiogenesis, you can also use the VEGFR2 mouse, and VEGFR is only expressed or used when there is new vessels that are developed inside tumors, for example, or just to recover from myocardial infarction or this kind of thing. So many different transgenic animals already exist, and many of them has been recently being developed by researchers, especially due to the fact that CRISPR-Cas9 really helped to insert a piece of DNA coding for luciferase, for example, in a specific position inside the genome of cells or animals. So different LPTA stands for light-producing transgenic animals that are emitting light when those pathways are activated. To show you an example, so this is the same animal on the three slides, but we are just having a look at three different events. So those mice have been grafted with tumor cells. So the tumor cells can be visualized by fluorescence. So we used a fluorescent protein. It's cherry, I guess, in this case, or TD tomato. So we could detect the tumor cells using these gene reporter techniques in fluorescence. Those mice were transgenic animals, and they are VEGFR2 transgenic animals, which means you can see here the host response. So the tumor cells will emit chemicals, and these chemicals will create new vessels to irrigate the tumor. So you can see here the host response. This is just the activation of the VEGFR2 pathway in the mouse. And we also used an antibody labeled with a dye at 750. We have injected the dye, and you can see the localization of the dye here in the tumor. So we are targeting the tumor with an antibody in fluorescence. We are just visualizing the tumor cells in fluorescence, and we can see the host response, especially regarding the creation of new blood vessels, so the neoangiogenesis that is occurring inside this animal. And all those three things can be just measured into the same animals. Just to give you some, to show you some images of different light-producing transgenic animals. So all those transgenic animals are used in inflammation. So whenever GAD45 beta pathway is activated, the mouse will emit light, or E-kappa B, or NF-kappa B, or SAA. So it's always models where we are injecting either PBS or LPS, for example. And here you can see after LPS injection, IP, you can see a huge increase of light from the peritoneal area. Here, the same E-kappa B, so we have injected a product that is inducing inflammation. The same thing in the PERIT one, and you can see the inflammation over the time. Same case here with two different compounds that are easily creating inflammation, CCl4 and sodium arsenite. And you can see that the mouse will have a peak of inflammation 7 hours after injection for sodium arsenite, and more 24 hours for CCl4. Okay, just to show you another approach using bioluminescence, we can also use bioluminescence to image a GPCR ligand binding directly in vivo by using a BRET technique. So BRET is bioluminescence resonance energy transfer. So those researchers developed a cancer cells that is expressing a beta-2 adrenoreceptor in fusion with a luciferase enzyme. So all the adrenoreceptor, especially the beta-2 one, are fused with a luciferase gene. Not a luciferase gene, with a luciferase. So when you are looking at those cancer cells, since all the beta-2 receptors have an enzyme grafted to it, if you have the substrate called propanolol with fluorescence that can be excited by the blue light emission from this luciferase, you can transfer the energy. So the luciferase will emit light, this light will excite the dye, and you will collect just the dye emission, so the fluorescence from the dye. So in the end, you can just detect the tumor cells by detecting all the beta-2 adrenoreceptors, because you can see the blue bioluminescence from the cell. And whenever there is a binding between your ligand and your receptor, then there is an energy transfer and you can measure light in the red range. So what they did, especially in this case, is they did some competition. So what you can see here is the red signal. So the red signal is the red signal, and the bioluminescent signal is just the localization of cells. So there is two kinds of group, control group and treated group. What they mean by treated group is that they are using a huge amount of the same ligand, so propanolol, but the competition is done by propanolol without dye on it. So here you can see the propanolol with dye. Okay, so you can see the light emission because this propanolol is really in touch with the beta-2 adrenal receptor. So this bioluminescent signal is transferred to the dye and you can find red light emission. If you add a huge quantity of non-fluorescent propanolol, you will see that there is no red signal, red transfer anymore because most of those receptors are filled with non-fluorescent propanolol. So they were able to perform this competition and to really prove that this fluorescent red signal is really coming from the presence of propanolol inside its receptor. So this is a very nice and imaginative approach to be able to see some protein-protein binding directly in vivo. Okay, this is another approach using, again, it's T cells. So it's T cells that have a specific construction. They have on one side one green luciferase with a very strong promoter. So the green luciferase is expressed all the time. And this green luciferase will be used in order to visualize the localization of the T cells. And at the same time, they have also another luciferase, which is emitting light in the red range. But this one is with a promoter which is expressed only when the T cells is active. So you can see with the red luciferase, the T cell activity, and with the green luciferase, just the T cells localization. So here you can see that the T cells are localized again in the lymphoid organ, the thymus, the spleen, lymph nodes. You can plot them, you can extract the organs, and you can make some ex vivo measurements. And the beauty of the study comes when you are using a vaccine. So people are using, in this case, two different vaccines, a control vaccine, which is not stimulating T cells at all, and another vaccine, which is an OVA vaccine, which is stimulating T cells. So you can see here in the green range, the localization of the T cells. So very few T cells were recruited in the area where the vaccine has been administered. And in the case of the OVA vaccine, you can see that there is way more T cells that is in the environment of the vaccine. And when you look at the red signal, this is the activity of the T cells. You can see that with the control vaccine, there is no T cells activity. But in the OVA vaccine, you can see that T cells are present and they are active as well. Okay, I would just like to finish with just not bioluminescence. I am done with bioluminescence. I have just 2 to 3 slides showing excuse me, a specific tool we have on our instrument regarding fluorescence. So you know that the main problem with fluorescence is what I call the bad photons. So in order to be able to remove the bad photons from the right photons, we are using what we call a spectral mixing strategy. So the spectral mixing strategy concept is to perform multiple acquisition with multiple different filter pairs. Okay, and the idea is to be able to separate dyes based on their spectrum. So this is just a mouse with like a specific signal somewhere. You scan your animal with multiple filters, and this is what is measured. It's just the sum of your specific dye and autofluorescence. So both of them are mixed, and the spectral unmixing algorithm is able to perform what we call a deconvolution, is able to understand that this curve here is just a mix between the red signal and the green signal. And it can extract only the signal which is coming from this spectrum, or photons that are following this spectrum versus photons that are following this spectrum. So in the end, after spectral unmixing acquisition, you get two different images. One image with only your reporter, this is the one you are going to use for quantification, and the second one just with the bad photons, the ones that are not useful for you. Just concretely, how does it look like? This is just the raw data. So the raw data are all the different filter pairs that we need to use in order to be able to measure spectrums. So this is the raw data. And as you can see, there is fluorescence everywhere. There is fluorescence in some specific areas, like the bladder or the tumors that have been injected into these mice. But there is also fluorescence everywhere in the mouth, in the skin, probably some food as well. And, well, we have injected two probes in this animal. And after performing the spectral mixing algorithm, we decided to create multiple maps. One map with autofluorescence, one map with just the first dye, one map with the second dye, and one map with just the food signal. As you probably know, food contains chlorophyll, and chlorophyll is really like a dye at 680 nanometers. So here you can see the spectrum from the food, spectrum from the dye at 680, and the spectrum from the dye at 750. And just based on those spectrum, we can really create maps with just your dye of interest separated from the other dyes. So same animal, and we can measure here the MMP sense. So this probe is just emitting light when it's cleaved by a metalloprotease, and this one is emitting signal when it's cleaved by catepsin. So you can see that both of them are shining from the tumor. So both tumors from both sides are synthesizing MMPs in the inflammatory environment, as well as catepsins. You can also see that there is a non-specific signal coming from the bladder. This is just coming from the clearance of the probe. You can see the food signal, which is not a good signal. We don't want to quantify this one. And you can also see that there is a skin signal, which is not displayed here, but there is also lots of signal from everywhere in the mouse. Just to show you a last example, so this is just a composite image. So you can see one signal in red, one signal in green, the other signal in blue. And when the red and the green signal are colocalized, it turns yellow. Last one, well, it's not exactly the last one, I just have a final one at the end. Just to show you that we can also quantify very accurately quantity of dyes, even if they are overlapped. So here we used two different dyes, one dye at 645, another dye at 680, and they are so close that both spectrums are overlapped. If you are not using any spectral mixing sequence, you will have to use just a single filter pair. So this is the single filter pair with the most optimal filter for the first reporter. And this is the filter pair acquisition, so with the most optimum excitation and emission filters for the second reporter. Here in this mouse, we have injected both dyes in different concentration. So we have injected increasing concentration of the dye at 645 and increasing concentration on the other way of the second dye. And both signals are in the same area because we have done co-injection of dyes. What we can see is that there is no way with a single filter pair to just visualize one dye or the other. We are always visualizing both of them. So, as I said, just because the emission spectrum is overlapped, if you are just using a single filter pair, there is no way to discriminate which signal is coming from which dye. But if you are using spectral mixing, you can first remove the autofluorescence from the skin. This is what we have done here. And you can ask for two maps. One map with just the dye at 645, and you can see that we can find this increasing concentration of the first dye. And you can ask for a map for the second dye, and again, you can find the increasing concentration of the second dye. Even if, because those mice are exactly the same mice, even if the signals were overlapped, we can detect what was the quantity of the first dye and the quantity of the second dye. Again, if you plot now the quantifications, so this is another study on different mice, but it's the same approach. Two dye, one dye at 645, one dye at 680, that have been co-injected with different concentration into the mice. So same as here, increasing concentration of dye 1, increasing concentration of dye 2, and this is the result with the quantification. We can see increasing fluorescent signal from 645, increasing quantification quantity of the second dye, and we can also see the food signal here in the stomach and in some intestine. So the only way to quantify your dye and to not quantify some food that goes with it, it's really to perform spectral mixing. This is the result, increasing concentration of dye 1. We are quite linear with a good 0.99, which is quite good. And for the second dye, the one at 680, we are also pretty good. When you increase the concentration, we have more and more light collected. So this is a good way, a good tool, spectral mixing, to be able to discriminate signals based on the spectrum. And I would like to thank you for your attention. And if you have any question, feel free to ask. I am ready for you.
Yeah, I think Carlos has sent us a question.
Okay, what is the
through
question?
the chat, yeah.
Yes, it's a patented algorithm, so we are the only one doing spectral mixing. So all the other people, they are just doing filter per acquisition. So you have to select your emission filter, your excitation filter, and that's it. And you collect whatever you have in this range. It could be autofluorescence, it could be your dye of interest, of course. But thanks to spectral mixing, of course, it's a bit longer because you need to acquire with multiple filter pairs, so it takes slightly more time. I don't know, a normal fluorescent acquisition is about a few seconds. Then it will take 5 times, 6 times, 9 times, a few seconds. So it's going to be 9 times longer, but the overall process is still very fast because fluorescence is fast. Is there any other questions? Spectral mixing can be done by your PerkinElmer equipment? No, only with PerkinElmer. Do you know of any Luc transgenic expressing specifically in the lung? So there is one mice which is not specifically expressing luciferase in the lung, but in all the different cells. It's the beta-actin Luc mouse. So this mouse is expressing luciferase in all the cells because beta-actin is always active, so it's a very strong promoter. So this transgenic animal can be used as a donor. So you can pick up from this mouse your cells of interest, T cells, macrophages. So I guess you can also extract from this beta-actin mouse some cells from the lung. But just one mouse expressing luciferase in the lung only, I have no idea of any of them. You can create your own. You just have to find a pathway which is only activated in the lung and not in any other organ. Or, for example, surface protein, which is only present in the cells from the lung, so the lung parenchyma. And if this specific surface protein is expressed only on lung cells surface, if you put a luciferase gene associated to the promoter of this protein, each time the specific membrane receptor will be expressed on the cells of your lung, you will have also production of luciferase. So this is something that can be done, but I have not in mind any mice which is spontaneously emitting light from the lung right away. So it has to be created. You can have a look in Taconic. So Taconic is hosting many different transgenic animals. So Taconic is like this. Taconic. You can find many of them there. And after that, it's like a partnership with people that are developing their own transgenic animals. So you just have to do some bibliography and to see which team has created which transgenic animal. And you can just reach them and ask if there is a way to do a partnership. Or if you know any Transgenose Institute, you can ask those people to create your transgenic mice for you. So, okay, thanks very much. Yeah, all the JAKs in all these mouse providers, they are not providing lots of luminescent animals. I think they almost have no luminescent animals. So, okay, if there is no other questions, I hope that at least you learn interesting things, that you have new strategies that are coming to your mind. The idea is really to just stimulate you on optical imaging and just to find maybe new ways to study what you are studying. So as you can see, you can really apply bioluminescence to many different applications. It could be used almost everywhere. It's a gene reporter technique. It's very sensitive. It's also linked to viability. So that's all the beauty of optical imaging, and especially bioluminescence.
Mm-hmm. Okay, thank you very much, Guillaume. Thank you, Carlos, for attending, and thank you, Teresa. Teresa de Francisco has been connected the last, I think, 30 minutes.
Okay.
Yeah, and she asked sorry because something has been
No
engaged
problem.
with her. Yeah.
As I said, I will share my presentation, so there is a lot of, I would say, content in this presentation. So you can still go again over it, and just, even if something was not very clear from my side, you can just go and have a look again into those slides.
Okay. Okay. Thank you very much.
You're welcome. Many thanks for attending.
Carlos,
Bye bye, everyone.
nos vemos. Hasta luego. Chao. Adiós, Teresa.